Hi, today we're going to look at another required practical for AQA GCSE Biology, and that is food tests. Now what that means is we're going to be examining these 5 mystery solutions and trying to find out what biological molecule resides in them. In here I've got random solutions I've asked our lovely technicians to make, and it's going to be, some are going to be glucose, starch, protein, or a lipid, which is another word for fat or oil. Okay, so we're going to use the different chemicals to figure that out. So like any good scientist, we have food samples A through to E. We also have the different tests that we're going to be running, and I'll talk you through the steps for each one for you guys to make note and to note down what are the different steps to making that. So let's get cracking. Let me get my safety goggles. I have here 5 mystery solutions, and we're going to take a look at each one in turn to see what they entail. So let's start off first with the Benedict's test here. We're going to run all 5 of my solutions through the same test. Off we go. So my first test is the Benedict's one. We're using Benedict's solution, which is a blue color, and that's of note, very important thing to note first. So let's very quickly just label all our test tubes, A to E, and then we're going to run the same test for all of them. Okay? We're then going to use a different pipette for each different solution so as not to contaminate them. So 1, 2, 3, 4, 5. I'm going to be as methodical as I can about this so as not to mix it up. So I'm going to use 1 milliliter of each one. Okay, so I've got my 5 solutions. I put 1 milliliter in of each one. I'm then going to put an equal volume of Benedict's into each one as well. Now, the trick about the Benedict solution is it requires heat for the reaction to occur. So, because we're going to use the Bunsen burners when you guys come back, really. So we're going to make it easy, and to keep it safe, really, we're also going to use just your typical water bath. So I'm going to pop the kettle on, and then I'm going to put each of these in there. Trust the thermometer to make sure it's over 80 degrees. Watch the kettle cook. Yeah, over 80 degrees is good. We should then have a positive result in about 2 minutes time. Right, let's prepare our next ones as well. The next one is the Starch Practical, and that's a bit simpler. We're going to put each of these on. I'm just going to label them. And then I'm going to put a drop of each solution onto that. And then I'm going to test and see, using iodine, whether starch is present. All we need for this one is a drop. So this is coming along nicely. We'll see a result very soon. Let's have a quick look at the iodine one. The iodine one is immediate and very quick, actually, so we should be able to see the result very quickly. So just for your perspective, that's A, B, C, D, and E. A drop of iodine into each one. No color change. Ooh, turned black, purple, black. No color change. no color change, no color change. It stays brown. So a positive result is when it goes from brown to blue-black. So we're going to put down the iodine that B turned blue-black. So that means negative, positive, negative, negative, negative. That was easy, wasn't it? That's still developing. We'll come back to the Benedict's test in a second. Our next test is the Biuret test. So that is a test for protein. So again, prepare my glassware for my 5 samples. So the Biuret test is quite straightforward. It is basically a solution made up of 1% copper sulfate and 0.4 molar sodium hydroxide in equal volumes. So let's say we use 1 ml for the samples, we're going to use 1 ml of each one. So let's put that in there, put the samples in. So equal volumes of both. We should then get a very bluish mixture. If the blue turns to lilac or purple, then we know we have a positive result. And here we have it for your very eyes. A nice clear positive result. I'm going to use the white background here. Nope, not useful. Not useful. White background, man. White background, that's all I need. You can see the white background and then the color. Yeah? So A is quite obviously purple. So A is positive for protein. Just put that down. Positive, and the others are all negative. So we know already A, protein, B, starch. Now, the results of my Benedict's tests are through. In my hot water bath, I see, aha, solution D has turned orange. All the rest have stayed blue. So, orange or brick red, depending on the concentrations of the glucose present. So we know that D is our glucose. Oh, no. No, 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 yes. No. To recap, one more time. Benedict's is a test for glucose. It goes from blue to brick red or orange, and that was in solution D. Iodine is a test for starch. It goes from orange, this time orange, or orange-brown, to blue-black, or a dark deep purple, and that is B. Biuret was blue. In the presence of protein, it turns purple, and that was for A. So now technically we can eliminate A, B, and D, and we're just trying to figure out between C and E now what the thing is. So let's go through. That's done. Let's now figure out between A and E which one is my fat. Not A, C and E. And one of them is water. So here we go. Now, the way to test for fat, presence of lipid or fat, is through what we call the emulsion test. So the way to test for an emulsion would be to simply put the liquids in. So C was our latest unknown. Step 1, add ethanol to your solution. So ethanol just helps to dissolve the fat because it's an alcohol. Just pour it in a little bit. I should have used the squeezy thing, but I forgot. Equal volumes. Okay, we're then going to shake it up a bit, make sure that all the liquids, all the fat does dissolve in it. We're then going to add water, and if fats are present, or a lipid is present, it will turn the water a nice cloudy white. So let's do it with C first. And we can see it's clear still, so there's nothing going on there. So that's a negative for that test. As for this one, it has turned a cloudy white precipitate, or emulsion rather, has formed. There we go. Positive for the emulsion test. So we know then that C is not, well, these ones I'm telling you that they're not. So E is our one. And there you have it. Food tests.